Hello everyone, welcome back to BioBoost. Today we will cover one another cell organelle, that is lysosome. So lysosomes are membrane bound dense granular structures which contains hydrolytic enzymes. Means this is a cell organelle which is also membrane bound. It is a single membrane bound. In this diagram you can see hydrolytic enzymes are present inside it. And origin of lysosome is from SCR and Golgi. Means lysosome is part of membranes of SER, smooth endoplasmic reticulum, and Golgi both. Means it gets budded off from these both cell organelle. Their size varies from 0.2 to 0.8 micrometer. The word lysosome, as you can see, is made up of two words, lysis, and which means breakdown, and soma, which we know means body. So lysosome, the first part lysis is breakdown and soma is body. Lysosomes occur freely in the cytoplasm and found in eukaryotic animal cells and plant cell. But these are absent in mature mammalian RBCs. This point you need to remember from exam point of view, that in mature mammalian RBCs there are no lysosomes. Lysosomes were discovered by a Belgian cytologist in 1955. Whose name was Duve, and he designated them as suicide bags, right? So we will come to know why these are called suicide bags, but this scientist, a Belgian cytologist who studies cells, so lysosomes were discovered by Duve in 1955. And he called them as suicide bags. They are also known as the suicide bags of the cell. When cell gets damaged, lysosome may burst and the enzymes digest their own cell. Means when cell is damaged, they burst and enzymes digest their own cell. They possess different kinds of hydrolysis on release of these enzymes that cause death of cell. So lysosomes are sphere-shaped sacs filled with hydrolytic enzymes, and they have the capability to break down many types of biomolecules. So this is the main definition of lysosomes. Now we study about lysosomal enzymes, the enzymes present inside it. So it contains acidic hydrolytic enzymes, means the pH is usually low, and almost about 50 of enzymes have been identified so far. Now all these enzymes are classified in 6 categories. 50 enzymes are there total, all are acidic in nature, and they have been categorized in 6 categories. First is proteases. The name itself suggests that it digests proteins, means the enzymes which digest proteins. Second is lipases, which digest lipids. Amylases, which digest carbs, carbohydrates. Nucleases, which digest nucleic acids. Phosphoric acid monoesters, and sulfatases. Now collectively the group of enzymes is called hydrolases, which cause cleavage of substrates by the addition of water molecules. And most of the lysosomal enzymes function in the acidic medium. Now these enzymes have a nature that they function in acidic medium only. So these 6 categories are important to remember. Now we will study the structure of lysosome. So The outer surface of lysosome is formed by a single membrane, which is basically a phospholipid bilayer that have some other membrane bound organelles also. So basically it is a phospholipid bilayer. Lysosomes are formed by budding off of the Golgi apparatus. Means, as I told you, the structure itself gets budded off from Golgi, and the hydrolytic enzymes within them are formed in the endoplasmic reticulum. Means the outer covering it's budded off from Golgi, and the hydrolytic enzymes present inside the lysosome are formed in ER or endoplasmic reticulum. Lysosomes are membrane delimited organelles. They also have a high concentration of protons, which results in pH which has a value of less than 5, and because of high proton concentration the pH is low, which is necessary for functioning of the hydrolytic enzymes present inside it, right? The surrounding membrane is composed of integral proteins as well as a vacuolar type H plus ATPase, which are basically highly glycosylated proteins, and a number of transporters. Presence of high protons helps in highly glycosylated proteins and a number of transporters to stay there. And depending on the type of lysosome and their function, they also greatly vary in size. They are less confined compared to other organelles. Next we will see types of lysosomes. So lysosomes are basically of 4 types, and that is why the organelle is known as polymorphic organelle. First we will see primary lysosome. In the diagram below I will show you these 3-4 types of lysosomes. Here you can see primary lysosome is shown from ER to Golgi, and Golgi it is budded off and made of primary lysosome. So they are formed from Golgi body appearing as small vesicles. So they appear as small vesicles and get budded off from Golgi. Primary lysosomes are popular on Golgi. They also occur as granulocytes and monocytes. Means primary lysosomes can be present on Golgi itself, and they occur as granulocytes and monocytes. They are basically surrounded by a single phospholipid layer and contain acid hydrolysis. A single layer of phospholipid. We know a mature lysosome is bilayer, but these primary lysosomes have a single phospholipid layer and they contain acid hydrolysis. Small sac-like structures enclosing enzymes synthesized by the rough RER. So they are simply called as storage granules, which are storing enzymes. Next we will study secondary lysosomes. So secondary lysosomes are formed of fusion of primary lysosomes with phagosomes, pinosomes and endosomes. Here in this diagram you can see phagosome is there and other pinosome and endosomes are there. So they are fusion of primary lysosomes with either of these 3 somes. They are making secondary lysosomes. Compared to primary lysosomes, secondary are larger in size and are capable of releasing their content, meaning enzymes, outside the cells where they degrade foreign material. Means their sizes, because they are made up of fusion with these 3 and primary lysosomes, so they are basically larger than primary, and they are capable to release their enzymes outside of the cell also to degrade any foreign material which has entered the body. So they contain active enzymes. They contain active enzymes and materials are progressively digested. Next is tertiary lysosomes. So tertiary lysosomes, these are also known as telosomes. In this diagram you can see the residual body, which you can see here, is basically telosome or tertiary lysosome near secondary lysosome. So these are basically residual of secondary lysosomes, and they contain undigested food which later on gets expelled by exocytosis, which is a process by which the unwanted material of cell gets eliminated from the cell, and this process is basically exocytosis. The last type is autolysosome. So autolysosomes are also known as autophagosomes. In this diagram you can see a autophagic vesicle, it is shown. So these are formed by fusion of primary lysosome with cell organelles which are old or damaged and need to be digested. So primary lysosomes is attached to cell organelle which are needed to be eliminated from the cell, so they fuse with these and make a autolysosome which get eliminated. Autolysosome also function during deficiency of food by digesting the reserved food. Means the food material gets itself reserved food which is present inside the cell get digested, and deficiency of food is settled. They are also called as suicidal bag when they rupture to release all the enzymes to digest and the old or worn out cell. So these are the type of lysosomes which are known as suicidal bags, because they rupture to release all the enzymes so that these enzymes digest the old or worn out cell. Means the old cells get digested, so they rupture themselves. Next we will see function of lysosomes. So the primary function of lysosome is intracellular and extracellular digestion at acidic pH. So the most important function of lysosome is basically intra and extracellular digestion at low pH, because it has acidic enzymes, hydrolytic enzymes, which work at only low pH. So different kind of digestion is there. First is intracellular digestion, which digest food. The lysosome membrane fuses with the membrane of food vacuole and squirts the enzyme inside. The digested food that diffuses through the vacuole membrane and enters the cell to be used for energy and growth. Right? Next is extracellular digestion. Here, primary lysosomes secrete hydrolysis outside by exocytosis, resulting in degradation of extracellular materials. Means in extracellular digestion, the primary lysosomes which are present, they secrete enzymes, these are hydrolysis, and release outside of the cell by exocytosis process, which result in degradation of extracellular materials. Means if cell needs to digest materials which are present outside of the cell, then extracellular digestion is performed by the help of exocytosis process. Next is heterophagy. In this diagram here you can see heterophagy